Hello, hi, welcome to the new lecture series of genetics. In this lecture series, we will be dealing about genetics, which is an important unit for the NEET examination. The unit is genetics and evolution, which is the seventh unit of your NCERT textbook. In this unit, there are mainly three chapters. First is the principle of inheritance and variation, and second is the molecular basis of the inheritance and variation, and third is the evolution, chapter evolution. Usually, 13 percentage in 2022 NEET examination 2023, I think so, about 13 percentage of the portions or the questions were from this unit. That means in the biology which have 90 questions, right? So it is a most important chapter for your NEET examination. In this chapter we about genetics, the basics of genetics. The first lecture we deal with will be, this is the first lecture, and this first lecture we will be dealing about the genetics, basics of genetics. So first of all, we will be dealing some of the important terms which are associated with the genetics. Then we will be moving to the advanced form. So I hope if you are writing it, if you are making your own notes with me, it will be better. I will not be giving any extra time for your note preparation. You can write the note with while I am taking the class. So certain important terms which are related with genetics. First of all, we will be learning what is a gene. What is a gene? Then alleles, then locus. Then we will be learning what is a chromosome, then chromatin, then homologous chromosome. Homologous chromosome. So in place of chromosome, I will be giving this hash sign, right? In place of chromosomes, I will be writing this hash. Okay. So some of the important terms we will be learning will be what is a gene, what is a allele, what is a locus, what is a chromosome, what is a chromatin, and what is a homologous chromosome, and homeologous. That is not needed. We will be writing it, by the way. Homologous chromosome and what are sister chromatids. These are the basic things you need to understand. I think you may have heard these terminology in your previous classes, right? So allele, gene, locus, chromatin, chromosome, homologous chromosome. So these are the terms which we will be learning. First, we will be learning about so what is genetics, right? So if you have noticed, if you have read the chapter name, principles of inheritance and variation, we are studying the two terms, inheritance and variation. We are studying its principle and the mechanism. So this chapter we will be dealing about inheritance and variation. In the next chapter, which is the molecular basis of inheritance and variation, we will be studying about the molecular basis, how the things are inherited. Some things are inherited, right? And there are some variation in the inherited thing. So we will be dealing about in the next chapter. And this chapter, third chapter, will be your evolution chapter, right? So once again, I welcome you everyone to this lecture series. So first about what is genetics, right? Genetics is actually the branch of biology. We have a varied branch of biology, right? So genetics is the branch of biology which deals with the inheritance and variation. So what is inheritance and variation? So first of all, we will be dealing about inheritance. What is inheritance? From the word itself, from this word itself, we can understand something is transferred from one organism to other organism, right? Or from the parent, and or we can say that something is transferred from the parent to the its next generation. So if we think of human beings itself, right? We human beings are, everyone are not same. right? We are not exactly same as our parents. We are not exactly same as, I am not exactly same as my father, or I am not exactly same as my mother. There are certain variation from my parents, or I can say that there is something inherited from my parents to me. So there comes the two things, inheritance and variation. Something is taken from my parents, something is not taken from my parents. The things which are not taken from my parents will be going to variation. Some things are taken from my parents, that's inheritance. So if I give a good definition of inheritance, what is inheritance? Inheritance is due to the heredity. So what is heredity? Heredity is the process of transmission of certain heritable characters from the parent to the next generation. So what is the basis of inheritance? Basis of inheritance is the heredity. And what is variation? Variations are certain differences which are seen in the progeny compared with their parents. So I will say the definition once more. What is inheritance? Inheritance is caused due to the heredity. By this, the traits are passed from the parents to the offspring. What is variation? Variation is difference which is seen in the progeny when compared with their parents. How variation is caused? Variation is caused due to the recombination during cell division, or variation can be caused due to mutation or environmental factors, etc. So, a good example of inheritance. I will give a good example of inheritance. Look, this is a good example of inheritance. There is three diagrams, right? Three diagrams of a year. If you analyze the structure of the year in this position, here there are three different years of three different persons, right? So we can see the difference. Some of them have their earlobe. This is known as the earlobe. Their earlobe is attached. Some of them, their earlobe is not attached, fully released, unattached form. But in some cases, there is an intermediate form. Why this arise? Why this arise? There is a question. This arises due to variation. Variation from them, either parents. Sometimes their parents will be having an attached earlobe, but their progeny or their next generation, some of their progeny will not be having an attachment, some of them will be having an attachment. These are the varied examples of the inheritance pattern which are seen. So they are not equal. There are three conditions. The fully attached form. This is the fully attached form, right? This is the recessive form. Recessive form means we will be studying later in this lecture series. What is a recessive? What is a dominant? And in case of this, the third picture, which is in your right, in here the earlobe is not attached. It is the dominant form. So these are the examples of inheritance and variation. Like, if just consider his father was having an attached earlobe, but for him his earlobe is not attached. So there is a variation, but maybe his mother earlobe was not attached. So this may be the condition which arises due to the inheritance pattern. So what is genetics? Genetics is the branch of biology which deals with inheritance and variation. Okay. What is inheritance? Inheritance is caused due to heredity. By this, traits are passed from the parents to the offspring. Some traits are passed. What is a trait? Trait certain character, or specific characteristics of an individual. For example, the previous diagram I mentioned, earlobe attachment. This is a trait. Some people will be having their earlobe attached. Some people will not be having their earlobe attached, and some people will be having an intermediate earlobe attachment. So this is the case of inheritance and variation. So we will be learning each and every terms now. So we have learned about genetics. What is the branch of genetics, right? There are various examples of inheritance pattern and variation pattern. There is no need to study this example. We are directly going to our topic. So, first we will be learning the terms each terms. Okay. So first of all, I am taking the case of human being. We know that human beings are deployed in condition. What is deployed in condition means? Deployed in condition means human beings, normal cells, right? Normal cells are deployed in condition, which means in each cell, right? We know that, consider it as a cell. This is a cell, and cell will be having its own nuclei inside, right? I will show a diagram, that will be better. Look, this is a cell, right? Cell will be having a nucleus inside it. Nucleus will be having its genetic material, which is the chromosome, right? So this is a chromosome. We will be studying the structure of chromosome later. Just look at the diagram. In human beings, we know that we are deployed in condition. What you mean by deployed in condition means, in each cell will be having a pair of chromosome, a pair of each chromosome. I think you understand what is a diploid condition. In a diploid condition, in each cell, a chromosome will be having a same copy of itself. So we know that in human beings we have how much chromosome? What is the chromosome number of human beings? Our chromosome number is 46, right? The chromosome number of human beings is 46, right? Okay. That means in human beings, if I am taking a cell, if this is a cell, if a cell is having chromosome, consider this as a chromosome number 1. So I earlier mentioned our chromosome number is 2n is equal to 46, right? It means we are having 46 chromosome. 46 chromosome means this 46 chromosome is seen as pairs, which means we will be having 23 set of chromosome. We will be having a 23 set of chromosome, which means chromosome 1, chromosome 2, chromosome 2, chromosome 2, chromosome 3, 3, 3, 4, 4, 5, 5, 6, 6, 7, 7. Like we will be having 23 set of chromosome. This chromosome is known as homologous chromosome, right? If, like, I will say that chromosome this is a homologous chromosome. What is a homologous chromosome? Homologous chromosome is the exact equal chromosome which is seen in a cell. Okay, this is the condition of a diploid organism. So I earlier mentioned we are having 23 pair. Chromosome 1 will be having its exact same pair. It is the particular copy of a particular chromosome. It is known as homologous chromosome. Okay, so that is the thing. Next, we have said what is a chromosome, what is a homologous chromosome, and next we will be discussing about gene. So I will be drawing a structure here. So we took the condition of a human being. We know that human beings are diploid in nature. Their chromosome number is in the form of 2n, which means a chromosome will be having an equal same chromosome in their cell. This condition is known as diploid cell condition. So in human beings we said that human beings are diploid, human normal cells are diploid in condition, but in case of human gamete cells they are haploid, right? Human gamete cells are haploid. After the fusion they will be forming a diploid zygote. These are the basic things that you may have known. So in human beings the chromosome number is 46, total 46 chromosome in a set of 23 sets. In that condition, I earlier mentioned we are having 23 sets. In that 23 sets, 22 pair of chromosomes are known as autosomes. 22 pair of chromosomes are known as the autosomes. Autosomes are known as the chromosome which are other than that of the sex chromosome. So what are sex chromosomes? Sex chromosomes are known as the allosomes. So we will be having two types of chromosomes, autosomes and allosomes. Autosomes are the entire set of chromosomes which are like 22 pair of chromosomes, which are other than that of the sex chromosome. Allosomes are the sex chromosome, which are X and Y chromosome. So if we take in, so we human beings are of two types, right? Human beings are male, we are having a male version and we are having a female version because we are sexually reproducing organism. Sexually reproducing organism need two parents, a male and a female, which will produce their own gametes, a male gamete and a female gamete, and they will fuse together forming a zygote. Zygote is the next generation. Thus there will be variation, right? Thus there will be variation. You should understand, in a sexually reproducing organism or a vegetatively reproducing organism there is no variation. We can't differentiate the male, we can't differentiate the parent and their daughter cells or their progeny. They are equivalent, they are exactly same, they will be show the exact same characters because there is no fusion of different parentage. There are no parentage, there is only a single parentage. The single parentage is transferred, the same things are transferred to the next generation. So there will be no inheritance. Sorry, there will be no variation. Usually it will be same. Okay. So if we are taking the condition of a human being, we know that there are two version, male and female version. So in case of a male, we will be having a 22 pair of autosomes. 22 pair of autosomes. 22 pair of autosomes plus we will be having X and a Y pair. X and Y pair. X and Y pair. So total this is a 22 pair. 22 pair which means 22 pair. It is a 22 pair which means 44, right? And other pair and one more pair. Pair means it is a two number, right? So 42 which implies which is equal to total number of 46 chromosomes. So total 46 chromosome, 22 pair of autosomes and a pair of allosomes which are the sex chromosomes. This is in the case of male. Usually male is denoted by this, right? It is in the case of male we will be having 22 pair of autosomes and X and Y. Okay, this is the condition in case of male. And in case of female there is a slight difference. The difference is they will be having a pair of X chromosome. A pair of X chromosome means they will be having two X chromosomes, so which will be having 46, total of 46 chromosome. Thus they will contribute a set of 23 set, right? This is the 23 set of chromosome. So this is the case of autosomes and allosomes. Then what is a homologous chromosome? I earlier mentioned homologous chromosome. It is the both the copies of a particular chromosome, both set of particular chromosome. So chromosome 1 and chromosome 1 will be homologous. Chromosome 1 and chromosome 20 will not be homologous. They are non-homologous chromosome because they have different in their gene structure. Whereas in case of non-homologous they are having difference in their gene structure. I will say what is a gene. Gene is only a part of a chromosome. I earlier have shown you a diagram. Okay, this is the diagram. You just look here. This is chromosome number 1. Okay, this is actually 23. This is a set of chromosome, 23 set of chromosome. That means each chromosome will be having a one more copy of itself. Chromosome 1 will be having its one more copy, okay, or in a normal cell, and chromosome X and chromosome Y will be having its own copies. So if I am taking chromosome 1 and chromosome 2 in a normal cell, this is the total number of cells. So I will be taking, just step here, I will be drawing a cell here, normal cell. Consider this as a, sorry, consider this as a normal cell. So in this cell I will be taking two chromosomes. Consider this as a chromosome number 1, and there will be the same chromosome number 1 will be present in the nucleus. Chromosome number 2 will be present in the nucleus. The same chromosome number 2 will be present in the nucleus. Chromosome 3 will be present in the nucleus. The same chromosome 3 will be present in the nucleus. Like in case of human beings, we can say that we have up to chromosome number 23, right? So we have a set of chromosome, and this chromosome will be have been contributed by your parent, one of your parent. This have been contributed from one of your parent, and this set of chromosome has been contributed by your other parent, which is your mother, maybe, right? So this is the case. Females are denoted by this diagram. Okay, so this set of chromosome is donated by your father, and this set of chromosome is donated by your mother, because they release gametes which are haploid in nature. Two haploid gametes fuse together to form a diploid gamete, a diploid zygote or a diploid cell. So this is the condition. So chromosome number 1 and chromosome number 1 will be homologous. Chromosome number 1 and chromosome 3 is non-homologous in nature. Why? Because chromosome number 1 and chromosome number will be having the exact gene, the same gene. What is a gene? Gene is a part of a chromosome which contains the heritable character. Okay, that is the condition. So we have said homologous chromosome, non-homologous chromosome, homologous chromosome. What do you mean by homologous chromosome? Homologous chromosome are the chromosome which contains homeo means it will be having certain similar structures, and also it will be having certain non-similar structures in their gene. In case of homologous chromosome, everything is equal. Two of them will be having equal genes. In non-homologous chromosome, the two chromosomes will be entirely different. But in case of homologous chromosome, they will be having certain similar structures. Okay, like X and Y. X and Y are the two different chromosomes. In case of female, it will be XX. In case of male, it will be XY. If we are taking in case of male, we will be having XY, right? So X and Y are homologous in nature because they are having, usually X and Y are different, right? They are different chromosomes, but in order for their attachment during the cell division, they are having certain common structures in their gene or in their structure, which are known as PAR, pseudo autosomal region. Pseudo autosomal region is the region in the in X and Y, so that they are the similar region, so they can attract. This is the case of homologous chromosome. Now we will be looking through certain other terminology, right? How much terminology we have gone through, right? Now we have learned, okay, we have learned homologous chromosome, etc. Now we will be going through much deep about the genes. So both the copies of a particular chromosome is known as the homologous chromosome. Now we are moving to next term, which is allele. First of all, we need to understand what is a gene. Gene is a segment of the DNA which have the coding information, and locus is the position of a gene in a particular chromosome. So here I am taking two chromosomes. These two chromosomes are homologous chromosomes. Just consider chromosome number 1, and this is also chromosome number 1, and they are homologous chromosomes as we know. And I am taking a gene for height. Here in this position at this locus, I am taking the gene for height. So if I am taking the gene of a height here, the same gene will be present in the same homologous chromosome. The next chromosome, the same locus. So, but we can't predict that whether the gene will be in its capital form or in small form. Okay, this is the gene which codes for the height of an organism. I'm just taking an example. So, what is an definition of an allele? Allele will be the alternative form of a gene. Alternative form of a gene which have the same locus on a homologous form. If I'm taking capital T, then if the small t comes in the adjacent or the next homologous chromosome, then the alternative form is known as the allele. This is the general definition for allele. Okay. Now, in a chromosome is actually a DNA. What is a chromosome? Chromosome is a highly condensed structure of a DNA, whereas chromatin is the less condensed form of DNA. So, one chromosome contains only one DNA, right? Okay. Next. So there is a common thing to remember that in case of a homologous chromosome, I'm taking an example, one homologous chromosome, a pair. So the concept of allele resides on the non-sister chromatids of a homologous chromosome. The concept of allele reside on the non-sister chromatids of a homologous chromosome. Which are the non-sister chromatids? So here, this both of them, this chromosome and this chromosome, this in this chromosome, this is the sister chromatid, and this is the sister chromatid of this chromosome. And what about non-sister chromatid? These two are the non-sister chromatid, or these two are the non-sister chromatid. Okay, that is the concept of non-sister chromatid. Now, actually, in a paired chromosome, what is a paired chromosome? In meiosis, or we know that in cell division, there are mainly actually two stages, right? The interphase stage, which consists of G1s and G2, and the next stage, which is known as the M phase. So, in G phase, in the interphase, we know that the interphase consists of G1, S, and G2. So, in the S phase, the chromosome gets multiplied into two, divided into two. That means in G1 phase, the chromosome will be like this, right? In the G2 phase, the chromosome undergo replication and forms a sister chromatid. The chromosome will undergo replication, and they will be having a pair of chromosomes which is attached to it. Okay, this is the case of replication. So, if a chromosome will be having a capital T, if one pair of homologous chromosomes, one chromosome will be having capital T, if it gets replicated after S1, sorry, after replication phase, the same T will be present in the sister chromatid, right? Same T at the same position, capital T will be present. Okay, so this is the case of capital T. What about the next chromosome? The next chromosome will be having small t. So after replication, the chromosome will be divided into two. Will be having an exact copy of exact gene will be here. This is t, small t, right? This is after the condition of the replication in S phase. Okay, so if we talk about the alleles will be We know that it is the alternative form of the gene. So here, the concept of allele will reside in the non-sister chromatids. These two are the sister chromatids, these are the non-sister chromatids. So the concept of allele resides on the sister chromatids of a homologous chromosome. It should be homologous. So this is an allele. Okay. So next we are going to see the next term. What is a genotype? Genotype. Genotype is actually a complete set of genes which are transferred from the parents to the offspring. The complete set of genes which are transferred from the parents to the offspring. So how this is transferred? This is transferred by the process of reproduction, right? In a sexual reproduction, the gametes have been fused and the genes have been transferred from the parents to the offspring. So there will be different condition of allele. If in a homologous chromosome, we are taking a pair of homologous chromosome. If in this loci capital T arises, in the same pair, if the capital T arises, then it is known as homologous dominant, because TT, there are, look, capital T, there is a chance that the both of the chromosome will be having the capital T. So this codes for the tall height. The organism will be tall. And in case of the dwarf, and the second case is that in first chromosome, it will be, the gene will be in capital, and in the same homologous chromosome, in the same locus, the chance may be, the T may be in small. How the there is a chance that the T becomes capital or T becomes small? It is according to their parents. It is by the fusion. We will be studying it very in detail in the coming lectures. So this may be the condition, and there is also a condition that only small t can arise. In this condition, the organism will be dwarf. The organism will be dwarf in condition. Sorry. Organism will be dwarf. So the next condition is that the middle one, right? The middle condition is that even though there is a small or a recessive form, negative T, but in usually the sexually reproducing organism, in this condition, the organism will be tall. So these are the three different combination of alleles. There can be only three different combination of allele if we are taking a single gene, right? We have taken a single gene which is a T, which is a gene for height. So this is the case of genotypes. So what this will be called as? This condition. This condition is homozygous condition, and it is tall, right? So it is tall, is the tall height is usually considered as a dominant character. So it is a dominant, homozygous dominant. If both of the T are in the small letter, then it is considered as homozygous recessive. And if one T is capital and the next T is small, then it is considered as homozygous, heterozygous or non-homozygous. Okay, non-homozygous, but it will be dominant. These are the three conditions. Okay, or we can call as heterozygous. Next, so this is the condition about genes, genotype. Next, we are going to study about phenotype. What is a phenotype? Phenotype, it is the physical form of an organism, or it is a physical form or an appearance or the observable character, right? Here there have been three different genotypes. What are the three different genotypes? Capital T, which is for homozygous dominant, and capital T small t, which is heterozygous dominant, and small t, which is a homozygous recessive. So there are here, the number of genotypes is 3, but here there are only 2 phenotypes. What are the phenotypes? The phenotypes are tall and dwarf. There is no intermediate character here. So here the genotype is 3 and the phenotype is 2. What are the phenotypes? The phenotypes are either tall, both tall and dwarf are the phenotypes shown by this organism. We have taken only a condition. This is case. There are the case may be different. Okay, that is the case of genotype. Genotype is the physical form or the observable character of an organism. Next, there is a question. So we should remember that genotypes always decide the phenotypes. Genotype decide the phenotypes. If the genotype of an organism changes, the phenotype also changes. The genotype controls the phenotype of the organism. We will be studying it in very detail in the coming lectures. This is only the basic class. Next, there's a question. If a genotype of an organism is given as capital A, the thing, how many different types of gametes would be formed? You should understand the question very carefully. Genotype of an organism is given. How many different types of gametes would be formed? So I will write. So I will write the genotypes here. Capital A, capital B, small b, capital C, capital D, small d, and small a. So if you are visualizing this, you can see how many gametes are, how many different types of genes are there. 1, 2, 3, 4, 5, right? A gene, B gene, C gene, D gene, and E gene. There are 5 genes. And what is the combination of A? The combination of A is in the A is in the dominant form, right? Capital A, capital A. But in case of B, it is also in dominant form, but it is in heterozygous form. Capital C, which is dominant, capital D, small e, which is in the heterozygous form, and capital E, which is in the its homozygous recessive form. The question is that how many different types of gametes can be formed? I will say the answer will be 2 raised to n, where n is the number of heterozygous gametes. N is the number of sorry, where n is the number of heterozygous. Where is the n is the number of heterozygous gametes. So here the answer will be 2 raised to how much? How many heterozygous gametes are there? There are only 1, 2. There are only 2 heterozygous gametes, right? So the answer will be 2 raised to 2, which is 4. So the total number, sorry, the different types of 4 different gametes will be formed. 4 different gametes will be formed. I will say how it has came. Look here, capital A, capital A. So for this, you need to consider a cell in its division. We know that in a sexual reproduction, the gametes are formed by the process of meiosis. Meiosis is actually the reductional division. So just consider 2 cells. This is cell 1. Sorry, okay. I will draw a chromosome here. There is a homologous pair of chromosome. Okay. So I am taking a gene here. I am just drawing a line. I am taking gene A. So according to this organism is having A in its homozygous form, dominant form or homozygous form. We can't say that whether it is dominant or recessive. And it is also having a B. It is also having a B gene, which is in its recessive heterozygous form. And it is having C gene, both are homozygous. And a D gene, which is heterozygous, right? Also a homozygous E gene. Okay, so in meiosis, I am just saying meiosis 2. What happens in meiosis 2? The cell will be divided into 2, right? Sorry for the inconvenience. So after meiosis, I am drawing, there will be, the cell will be divided into 2 and the chromosome will be separated. So here, so here the cell will be divided, right? So in this cell, capital A, capital B chance is there, right? And capital C, capital D, capital E, right? And in this cell, it will be capital A, small b, C, D, and E. Look, here you can see two equal cells are formed, but the gametes while meiosis have been splitted. So, according to our question, look, if I am taking a cell with only a single gene, just taking, I am taking an example of a single gene B. Here it is B and here it is small b. After meiosis, a cell will be containing a gene with capital B and a cell will be containing, this is gametes, right? So two different gametes have been formed. That is the thing which is being asked in the question. It is not the number of gametes formed, it is the number of types of gametes. So here there will be no different gametes. Here there will be no different gametes. Here there will be no different gametes. Here will be two different gametes formed. Here there will be two different gametes formed. So 2 raised to 2 is equal to 4. That is the answer. If so, I have earlier written equation 2 raised to n, right? That the number of different types of gametes, not the number, it is the different types of gametes is equal to 2 raised to n, where n is the number of heterozygous gametes. So this is the condition. It is not the number, it is about the types of gametes. So four types of different gametes will be formed: capital B, small b, capital D, small d. So two different gametes, two different types of gametes, four different gametes will be formed. This is the condition. So if you are given a large genome, if you are given a large genome with many number of genes, you should find the heterozygous, you should count the heterozygous gametes and you should put the number n in the equation. So actually this is the end of the basics of genetics. In the next coming lecture, we will be studying about the classical genetics. It is the basics. This is the basic of the classical genetics. In the sense, every genetics, the basics. So we will be in the next lecture, we will be studying Mendelian law of inheritance and the limitations, certain limitations are also there. So thank you. I hope you like my lecture and you should watch the upcoming lectures too. Thank you.